of RANI. Now, let us see how the food will get digested in the RANI. So, that is the process of digestion. The first and foremost question will come, what is digestion? Okay. So, whatever may be the organism, the food which is taken by the organism consists of different components like carbohydrates, fats, proteins, water, vitamins, etc. So when you come for the water or vitamins or any sort of mineral salts, all these are micromolecules. Okay, water, vitamins, mineral salt, all these are micromolecules, so which can be directly absorbed by the body by a phenomena of diffusion. Okay, but what about these carbohydrates, fats and proteins? These are macromolecules, and these macromolecules cannot be diffused directly. So first of all, they should be broken down, or they should be converted into simple micromolecules. That conversion of macromolecules into simple micromolecules is only called as digestion. In a more specific way, I can say conversion of complex, non-soluble, non-absorbable and non-diffusible. What is that? Conversion of complex macromolecules which are non-soluble, non-absorbable and non-diffusible into simple micromolecules which are soluble, absorbable and diffusible is known as the digestion phenomenon. What is that? The conversion of complex macromolecules which are non-soluble, non-absorbable and non-diffusible into simple micromolecules which are soluble, can be absorbed and can be diffused is called as digestion. And this conversion will be catalyzed by various enzymatic reactions. So with the help of enzyme, that entire reactions will be catalyzed and converted into simple micromolecules. So now you know what is digestion. So let us see how the digestion phenomena is occurring in our rabbit. So what is this rabbit? Rabbit is a herbivore mammal. So it obviously feeds on leaves, roots, tubers and vegetables like carrot, spinach, etc. So this is feeding on this various herbivorous food material, and that food material is complex, having the fats, carbohydrates and proteins. These all will be enzymatically hydrolyzed by various types of enzymes. Okay. Now the digestion phenomena in rabbit includes mainly three types. What is that? Mechanical digestion. Number 2, chemical digestion. Number 3 is microbial digestion. Coming to first one, mechanical digestion. What is this mechanical digestion? It is the phenomena which takes place in the mouth cavity. With the help of the teeth, the food will be masticated. That mastication of food by the teeth in the oral cavity and as well as the peristaltic peristaltic movements. You know very well, some muscular movements will occur in the alimentary canal, due to which the food will be passed on from one part to the next part of the alimentary canal. So this peristaltic movement and as well as the mastication method together known as mechanical digestion. Then what is chemical digestion? Whatever the chemical reactions that are occurring with the help of enzymes, so that are all hydrolytic reactions. That means they will occur in the presence of water. That will be called as chemical. And I told you already, in the alimentary canal of rabbit, there is a structure called as cecum, which is having the symbiotic bacteria. And that symbiotic bacteria helping in the digestion of cellulose. So because it is occurring by the bacteria, we are going to call it as microbial digestion. Okay, now let us see how the food will be entering into the alimentary canal and how it is getting digested in various parts. So first of all, with the help of its front legs, the rabbit will hold the food material like carrots or any fruits or any sort of leaves, and it will be taking into its mouth. And you know very well, it is having the incisors, premolars and molars. With the help of this teeth, the food will be masticated in the oral cavity. When the food is getting masticated, with the help of the tongue, the food will be mixed up with the saliva, which is produced by the salivary gland present in the oral cavity. Because the food is mixing up with the saliva, it will be converted into salt. That will be called as bolus. Now, how the digestion is occurring in buccal cavity? What is the first part? From mouth, the food will enter into buccal cavity. In the buccal cavity, the food will be mixing up with the saliva. And we know very well, saliva is having one enzyme called as ptyalin. What is that other name? Salivary amylase. So, in the presence of chloride ions, this ptyalin or salivary amylase enzyme acts on starch. Starch is a carbohydrate. So, on starch, this ptyalin or salivary amylase enzyme, which is present in the saliva, will be acting and converting into dextrins, and dextrins will be converted into maltose. In that way, some partial digestion of carbohydrate will start in the buccal cavity. Okay, right? What is happening? Simply, the ptyalin enzyme, which is present in the saliva, acting on starch and converting it into dextrins and maltose. Right? Now, from the buccal cavity, the food will enter into the esophagus. In the esophagus, there are no digestive glands, so simply it will pass through the esophagus and enters into the next part of the alimentary canal. What is that? Stomach. So let us see digestion in stomach. And you know very well, stomach is having the various parts like cardiac, fundic and as well as pyloric. So when you come for the digestion in stomach, if you have remembered, we said cardiac, fundic and pyloric gland are having the gastric gland and producing the gastric juice. Okay, do you remember? Gastric juice is produced by which glands? Cardiaca, fundica, pylorica. Gone with the wind? Okay. So cardiac and pyloric will secrete the mucus, whereas the fundic, the middle one, that will be producing the gastric juice. Okay. And do you remember what are the components of gastric juice? No? Okay. So gastric juice is having a acidic pH with 1.5 to 2, which is having the various components like HCl, pepsinogen. Now you will say, you will recall action of HCl. Number 1. HCl is strongly acidic in nature with a pH 1. So, because it is acidic in nature, the first and foremost function what it will do, it provides acidic medium to the food. What is that? Provide the acidic medium to food. Second function, because it is a strong acid, automatically any microorganisms are entering through food, that will be killed. So it kills the microorganisms present in the food. Okay, number 3. Number 3 is the pepsinogen is inactive enzyme. So HCl will convert the inactive pepsinogen into active pepsin. That's the third function. So pepsinogen, which is inactive, will be converted into pepsin, which is active. By whom? By HCl. That's the third function. And what is the fourth function? Prorenin. This is also inactive. This prorenin is also getting converted into active renin by HCl. This is also inactive. And last but not the least is it stops the action of thiolin. It stops the action of thiolin. We said it is mixing up with the saliva. Saliva is having the thiolin enzyme. So once the food enters into stomach, there is no need of thiolin action. That's why it will stop the action of thiolin enzyme. In that way, these are the functions of HCl. One is providing the acidic medium. Second one is killing the microorganism. Third one is converting the inactive pepsinogen to pepsin and prorenin to renin and stopping the action of thiolin. Now let us see the pepsinogen. As I told you earlier only, pepsinogen is inactive. So first of all it will be converted into active pepsin. By whom? By HCl. Okay. Now this pepsin which is getting active will act on the protein. In the acidic medium, they will be acting on the protein and converting them into peptones and proteases. Peptones and proteases. Okay? Proteins are getting converted into peptones and proteases by pepsin enzyme. Then, after the pepsin, it is about the prorenin. As I told you earlier only, prorenin is inactive, getting converted into renin by HCl. And this renin enzyme is present only in the infants, that is small babies. You cannot see the renin enzyme in the adults. Why? Because this renin enzyme is acting on the milk protein. And you know very well what is the food of infants? Milk only. So that milk will be converted, because in the milk there is a protein called as casein. What is the protein present in the milk? Casein. So this casein will be converted into para-casein by renin in the presence of calcium. Okay, remember, in the presence of calcium ion, the milk protein casein will be converted into para-casein, and this para-casein will be acted by pepsin, and it will be converted into proteases and peptones. And let me say, this para-casein on mixing up with the calcium is forming the calcium para-caseinate, which is a curd-like structure, white precipitate, which is a curd-like structure. And that's why you can see in the small baby babies, small infants, months babies, when they vomit, what they will be vomiting? They won't vomit the milk. They will be feeded with milk, but when they are vomiting the curd-like structure. Why? Because this renin enzyme is converting the milk protein called casein into para-casein, which is a white precipitate that is in the form of curd. So in that way, pepsinogen action, prorenin action. So please remember, renin is present only in the infant and it is not absorbed with the adults. Okay? And the last but not the least, what is that? Case's intrinsic factor. About this, already I have told you in the alimentary canal that the Case's intrinsic factor will be helping in the absorption of vitamin B12. Vitamin B12 is also called as cyanocobalamin. What is that? Cyanocobalamin. So vitamin B12 will be absorbed with the help of Case's intrinsic factor. Thus, in the stomach, because of the action of gastric juice, the food will be partially digested, converted into somewhat semi-solid food-like structure. So that partially digested, semi-solid or semi-liquid-like acidic food in the stomach is called as chyme. What is this chyme? Chyme is the partially digested, semi-solid, or you can say semi-liquid, semi-solid acidic food in the stomach. Please remember, chyme is important for very short answer question. So what is chyme? Partially digested, semi-solid acidic food in the stomach is called a chyme. So from now onward, what will be referring? We won't say it as food, we won't say it as bolus, but we will say that as a chyme. Now, after the formation of chyme, the chyme will enter very slowly from the stomach into the next part of the alimentary canal. What is the next part of the alimentary canal? Intestine. So let us learn about the digestion in the intestine in the forthcoming class. Now let us see the digestion in intestine. As I told you earlier only, the partially digested semi-solid acidic food in the stomach is called as chyme. As the chyme enters from the stomach into the duodenum part of small intestine, from the wall of the duodenum, different hormones will release, and that hormones will influence the liver, pancreas and intestinal glands. Because of that, bile juice will be liberated from the liver, pancreatic juice will be liberated from the pancreas, and intestinal juice will be released from the intestinal glands. Now, let us see how the proteins are digested, how the fats are digested, and how the carbohydrates. First one is, let us see, the digestion of proteins in intestine. So here, the pancreatic juice is having the various enzymes we have already discussed. Like what are the enzymes in the pancreatic juice? Trypsinogen, chymotrypsinogen, carboxypeptidase, amylase, nuclease and lipase. So here especially trypsinogen and chymotrypsinogen plus carboxypeptidase, these three enzymes will act on the protein. First of all, trypsinogen is a inactive be converted into active trypsin by enterokinase. What is this enterokinase? This is present in the intestinal juice. I told you already, chyme will be mixing up with all the juices produced by all the pancreas, liver and intestinal juice. So this enterokinase will convert the inactive trypsinogen into active trypsin. Now this trypsin will act on chymotrypsinogen, which is also inactive, and convert it into chymotrypsin. Okay? So trypsinogen is getting converted into trypsin. Chymotrypsinogen is converting into chymotrypsin. Now these two enzymes, what these two enzymes? Trypsin and chymotrypsin. These two enzymes will act on the proteins. Now you may ask me, madam, already proteins were digested. They are not completely digested. There it is the acidic medium. But here, as the food is entering into the duodenum, all this bile juice, pancreatic juice and intestinal juice, all these three are alkaline in nature. So the food will be converted into alkaline. So in the alkaline medium, the proteins will be acted upon by trypsin and chymotrypsin and converted into peptones and proteases. And all these peptones and proteases, this is equation 1, this is equation 2. All these peptones and proteases, two enzymes will act. What are those two enzymes? Carboxypeptidase and aminopeptidase. If you have remembered, carboxypeptidase is present in the pancreatic juice and aminopeptidase is present in the intestinal juice. So these two will act on the peptones and proteases and convert them into tripeptides, or you can say, polypeptides. And these polypeptides further will be converted into tripeptides and dipeptides. By the action of this carboxypeptidase and aminopeptidase, polypeptides will be converted into tripeptides and dipeptides. Then what will happen? Third one. Tripeptides will be acted upon by one enzyme called as tripeptidase. Do you remember where this is present? In the intestinal, and convert them into dipeptides. And dipeptides will be acted upon by dipeptidase enzyme present in the intestinal juice and convert them. Dipeptides will be converted into amino acids. And we know very well the ultimate end products of proteins will be the amino acids. Okay? So thus the proteins will be converted finally into amino acids. So thus the proteins will be getting digested in the intestine. Okay? So you have to write all these equations in the exam. Remember them in the form of equation, then it will be easy to write down the sentence form also. Okay? Trypsinogen converted into active trypsin, chymotrypsinogen converted into active chymotrypsin. Both will act on proteins, convert them into peptones and proteases. The peptones and proteases will be acted upon by carboxypeptidase, that will be converted into polypeptides. Aminopeptide will act on polypeptides and then convert them into tripeptides and dipeptides. And tripeptidase will act on tripeptides, then they will act on dipeptides. So in that way, totally proteins will be converted into amino acids. Now let us see, is that clear? Any doubts? Right. Now let us see the digestion of fats you have remembered, so far the fat digestion is not yet begin. Okay. Now coming to digestion of fats. So first of all, the fats which are large macromolecules, by the action of bile juice, they will be converted into emulsified fats. I told you bile juice will help in the emulsification. What is emulsification? Simply breaking down into simpler small molecule. This is the first step. Next, what will happen? On this emulsified fats, pancreatic lipase will act. Okay? Pancreatic lipase will act on the emulsified fat and convert them into triglycerides and diglycerides. And the third one, on this tri and diglycerides, who will act? The lipase of intestinal juice. The lipase which is present in intestinal juice will act on tri and diglyceride and convert them into fatty acids and glycerol. And we know very well what is the end products of fats? Fatty acids and glycerol. So thus fats will be completely digested into simple micromolecules called fatty acids and glycerol. Okay. Now let us see the digestion of carbohydrates. And if you have remembered, we have seen already the starch is converted into dextrins and maltose and all that. Now what will happen? Because of that dextrins and maltose, these all will be acted upon by various disaccharide enzymes, because these dextrins and maltose will be converted into various disaccharides like sucrose, maltose and lactose. Sucrose, maltose and lactose. Now, maltose will be acted upon by one enzyme called maltase and converted into glucose and glucose. Second one, sucrose. On this sucrose, one enzyme called sucrase or invertase will act and convert it into glucose and fructose. And third one, lactose. Lactose will be acted upon by one enzyme called lactase and converted into glucose and galactose. These are all disaccharides, whereas all these are monosaccharides. So like that, the disaccharides will be finally converted into monosaccharides, which are the end products of carbohydrate. So in that way, the complex fat, proteins and as well as carbohydrate will be converted into simple micromolecules. Like what? Proteins converted into micromolecules called amino acids. Fats are converted into micromolecules called fatty acids and glycerols. And our carbohydrates are converted into simple monosaccharides like glucose, fructose, galactose, etc. In that way, the digestion phenomena will be complete. Now, all this digested food material, of course, I told you already, water, vitamins, and mineral salts will be absorbed by simple diffusion. Now, the remaining, after the complete digestion, there will be a phenomena called absorption. So, the glucose and amino acids, they will be simply absorbed by diffusion phenomena, that is by active transport. Whereas the fatty acids and glycerol, they will be absorbed by villi, which are present in the small intestine. So in the small intestine, the innermost mucosa layer will show some finger-like projections called as villi, which will be increase the surface area of absorption. So these villi will absorb the fatty acids and glycerol, and thus the absorption also will be finished. Then whatever the undigested food, here the undigested food in rabbit still will be having some useful essential vitamins and cellulose, which is digested in the cecum cell. So that cellulose part and some sort of vitamins will be still left over in the fecal matter. So such fecal matter will be expelled outside by the rabbit during the night time. Okay, and this expelled fecal matter which is occurring during the night time, because it is having essential vitamins and some nutrients, what it will do, that rabbit will eat its own fecal matter. Why? It don't want to lose all such vitamins, minerals in the fecal matter. So during the night time, it will feed on its own fecal matter. So such phenomena is called as autocaprophagias. Auto means cell, caprophagias. So the organism which will eat the fecal matter will be called as caprophagias. What is that? Organism which will eat the fecal matter. For example, pig is there. So like that they will be called as caprophagias. But here it is eating its own fecal matter. That's why it is called as autocaprophagias. Now what will happen? Night time it is eating that fecal matter, and in the daytime, in a regular way when the food is taken, along with that, that undigested food also will be getting digested, and the only purely unwanted fecal matter will be stored in the rectum for some time. Later on that will be expelled outside during daytime. So the fecal matter which is expelled outside during daytime is the purely waste material, but that which is expelled during the night time is having some essential vitamins and minerals. So to avoid that one, it has adapted the autocaprophagias method. So this is the physiology of digestion in rabbit, which is very important for essay question, or sometimes digestion in stomach can be asked as separate short answer question, or digestion in intestine also can be asked as a separate short answer question. So either of the way,